Thank you for joining us for this poster. Low molecular weight component of almonds is cytotoxic to gastrointestinal cancer in culture. This is a study done at Bob Jones University. This is done by primarily undergraduate students. The first four on our list here, Evan Becker, Luke Brown, Emily Schwager, and Rachel Westphal, are all undergraduates who have since actually graduated from Bob Jones and are off going to grad school or working in the biomedical field. I am Dr. Steve Feigard. I am in charge of the Cancer Research Lab at Bob Jones University. And just to give you a quick little background there, we do have an undergraduate research program at Bob Jones, and part of that is the Cancer Research Lab that I'm in charge of. We have a summer program we call Research Immersion for undergraduates, which is where this research was done. This is an 8-week program during the summer when the students can come in and focus all their attention on doing research. And they, as we will see in a moment, we have found some component of almonds that seems to be cytotoxic to, well, actually is cytotoxic to cells in tissue culture that are cancer cells. So our background, as I said here, previous studies in this lab, we've demonstrated in vitro cytotoxic effect of almonds. We have been using 2 primary cell lines that I'll get to in a moment. And this current work in this poster confirms and further characterizes the observations that we've had in the past in this area. Basically, we take almonds and throw them in a Waring blender with some buffer, and we grind them up. We put them through a pseudodigestion procedure in which we adjust the pH and add some ingredients to try to mimic going through the stomach and the intestines. So we're trying to create a physiological type almond extract. One of the new things we did in this work is to run them through some molecular weight cutoff filters so that the material that came through would only contain components of a certain molecular weight and below. We also did a polyphenol extract trying to essentially determine what is the component of almonds that seems to be involved in this cytotoxic effect. Our primary method is using a dye called WST to measure the viability of cells in our ELISA plate wells where we're growing them. This dye is a dye that whenever cells are alive, they take the dye in and digest it or metabolize it into a chromophore that can then be read in a plate reader using absorbance. And so the higher the absorbance reading, the more live cells we have in the well. So we've been using a human gastric adenocarcinoma called AGS, a human colorectal adenocarcinoma called LOVO, and a human colon epithelial cell line that is a normal cell line. It's a little bit more difficult to work with, but we have been using that as a control nonetheless. So what we've found first in the colon cancer, or excuse me, in the stomach cancer, figure 1 here, we have AGS treated first with just PBS, and that it would be our control, showing 100% viability, and then that was used to calculate the percent viability in the remaining. And we have 5-fluorouracil as a as a positive control where we would see what cell death looks like in this system. Then we have increasing concentrations of this almond extract added to the cells. That's not 100%. It's 100% almond is not 100%. There is some media in the well with the cells so that they're not totally exposed to just the almond extract. And then we have 2 molecular weight cutoffs, 100,000 and 5,000. And you can see that we're getting equivalent cytotoxicity with the 100% almond and then with the 5,000 and the 100,000 molecular weight. So whatever is cytotoxic here appears to be a molecule that's 5,000 or less Daltons in weight, which probably eliminates most proteins. So it's probably, at least this is what we're thinking, it's probably more likely something like a polyphenol or a flavonoid or something like that. So that would be the first results we see here. The second results, this would be, sorry, we went to the normal cell line first. And please notice that the range here, those look like very wide, but we're actually only going down to 60% viability there. And 5-fluorouracil is not as effective in killing normal cells, which is a good thing because it's actually used as a chemo agent for treating some cancers. But you can see PBS and 100% almond are approximately the same, and the small molecular weight, probably within experimental, is equivalent. It's slightly higher in the percent viability range there, but it's not, if anything, it would say almonds are supporting cell growth relative to just plain PBS. Which is good because these are normal cells, and our interest is in seeing what kills cancer cells only, so that we can move on from there. Our third figure is our LOVO data. You can see we did do the polyphenol extract with both LOVO and AGS. I just didn't bother creating or putting in that graph as well, since we have limited space. But we again have PBS and 5-fluorouracil as controls. And then we have 100% almond extract, the low molecular weight, less than 5,000, and the polyphenol extract. And so we can see all those the almonds are giving us a good cytotoxicity that we're seeing there. So our conclusions then would be that there is some kind of anti-cancer agent or agents present in the almonds, and it's most likely a small molecular weight component, possibly polyphenols, but we have not fully identified that. And so our future efforts are going to be to try to identify what components of almonds are in fact responsible, and then try to determine the pathway that's being triggered for initiating the cell death in these cells.